Hello guys, I am Shubham from ShubhVed Tutorials. This video is about DNA replication. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. So basically DNA replication, what is it? A replication of DNA means DNA copies are being produced inside a cell. After a cell division, each cell should get the exact same copy of the DNA. That is very important, right? So this is a cell, right? After division, it becomes this one, right? Again, after that, this, this, right? Thus, each cell should have the same DNA content, right? Genetic information. Thus, DNA replication is required. Okay. So DNA replication before start, there is a cell cycle G1, S phase, G2, and M phase. These are the four phases, right? So S phase is concerned with DNA replication. The DNA replication takes place or occurs in S phase of cell cycle, right? So let's understand some of the important points of DNA replication. This is a DNA strand, okay? We know double-stranded structure of DNA, and these are anti-parallel. So first one is 5' to 3', another is 3' to 5', and these are complementary to each other. There are hydrogen bonds in between, okay? Anti-parallel, then complementary. Okay, they are complementary to each other means base pairing. What we can say? Thus, they are double-stranded in nature, right? They are complementary and anti-parallel. So, let's start with DNA replication. What happens? Just an overview. So first, there is formation of replication fork. Formation of replication fork. So what is the replication fork? For easy understanding, I will just draw here two strands of DNA, right? And these are bound to each other. Okay. So one is 5' to 3'. Another strand is 3' to 5', right? So after what happens? A replication fork is a structure in which this becomes a bubble-like structure. Okay, means here replication has started to occur, right? Again, 5' to 3' and 3' to 5'. Here are the bonds. Here the separation of bond has occurred. We can also say melting of DNA has taken place. Okay, also it is known as replication bubble. Okay, replication bubble. So this is known as replication fork because this structure resembles like a fork-like thing here and here. Right? Means fork means what we can say structure like this. Right? So this is a fork-like structure. So this has formed as the replication fork proceeds. See, DNA replication will occur in this direction as well as this direction. Okay, when in the it is the case of bacteria, right? Okay, so bacteria have circular chromosomes, right? Double stranded structure I will draw here. Okay. So when there is initiation, there will be a formation of bubble here like this. Okay, this is a bubble. Then replication will proceed in this direction as well as in this direction. So two strands will be formed. Right. Second point in the DNA replication is the leading strand and lagging strand. Again, we will draw a replication fork here. Okay, we will keep a place. All right. Now, see, we will assume again this is a 3 prime to 5 prime and 5 prime to 3 prime. First of all, we have to understand that DNA replication occurs in 5 prime to 3 prime direction. Okay, 5 prime to 3 prime direction means 3 prime OH, free 3 prime OH end is required for DNA replication initiation. Right. So, see, there is strand known as primer. Okay, primer which provides 3 prime OH. And this primer provides 3 prime OH. This primer is very important for the initiation of DNA replication. Right. So, I will draw here a short adhesion in the red color. See, again, this is a direction. The next strand complementary will be synthesized 5 prime to 3 prime. Alright. So here is a template. A primer, sorry. This is a primer. Okay. I will write here primer. Primer have complementary sequences. Okay. That will pair with the template. Okay. Both the strands will act as template. Template means they will provide the detail which nucleotide should be added. Alright. So here are the bonds. Like this, they will bond. Primer will bond. Primer is nothing but a RNA sequence. Okay, it can be 10 nucleotides, just 10 nucleotides. So this is the RNA sequence basically, and this will have a 3 prime OH group. So this is a 5 prime, and this is a 3 prime. So the growth will be in this direction, right? So this replication fork will open slowly, slowly, and here it will be complementary. Here will be the primer, right? And here the synthesis will occur. So this is 3 prime, 5 prime. These are the bondings. Now we will see a next picture in which the replication fork will go some nucleotides. Suppose I here. Okay. So after few minutes, this will be the picture. Replication fork has proceed, right, and replication is continued. So now more nucleotides have been added, right? So you can see here one strand. See, this strand is continuous strand. Okay, this will go in this forward direction as it is. But this strand is not continuous, right? This is not continuous. This is having a multiple primers, right? Because here is a replication fork till here only. Okay, from here, as the replication fork is added, new primer will be added, and then synthesis will occur. Because this is opposite to the replication fork direction, right? So direction of replication is this, and this strand is having the opposite direction. So the opposite strand is not continuous. Okay, and the strand which is following the direction of application fork is the continuous strand. So what we can write here, one strand is continuous. Okay, I will write here. So this strand is continuous, continuous strand, and another strand is non-continuous, non-continuous strand, right? So continuous strand is known as leading strand. Leading strand. And the strand which is not continuous, that is known as lagging strand. These are the important, what we can say, characteristics of the DNA replication. Lagging strand have fragments. We can see there are short fragments. This is the first fragment. This is the second fragment. As this replication fork will open, the another fragment will be formed. Alright. So this short fragment will be formed. Okay, which are joined afterwards after replication completion. They are joined. Primer removal takes place. Okay, primer is removed. Okay, because primer is just used for 3' OH. Right. It is removed afterwards. So these fragments of lagging strand are known as Okazaki fragments. Okazaki fragments. Now we will see the enzymes important for DNA replication. Okay, enzymes. First is the DNA polymerase, very important, which do the polymerization of DNA, right? Main enzyme in the DNA replication is the DNA polymerase. The DNA polymerase are of different types, type 1, type 2, type 3, type 4, type 5. All are important for the DNA replication thing, or we can say everyone have their different functions. So these are very important enzyme, DNA polymerases. Okay, polymerases from the name we can understand this is polymerizing enzyme. Okay, add nucleotides. Right, nucleotides are added. As we know, so what happens in the DNA replication? First of all, DNTPs, nucleotides are added. Okay, we require DNTPs. D stands for deoxyribonucleoside triphosphates. Okay, means, see, as I said, this is a 3' OH of a primer, right? And this is a ATP, suppose. Okay, means 3 phosphates are there. This is known as alpha, this is known as beta, this is known as gamma, right? So this 3' OH of the primer attacks the alpha phosphate. Okay, this alpha phosphate, and then the structure become like, suppose this is a growing chain, then this will attack adenine with phosphate. Okay, so this adenine phosphate will go there and this will be released out as 2p. Okay, so this is a basic reaction. Okay, and this is known as formation of phosphodiester bond. Right, like this another nucleotide will come. Suppose GTP, right? GTP. Okay, then again this will have 3 phosphates. So 2 will be removed out. G along with 1p will go with adenine. Right here. Okay, so this is the overall thing, a chemical reaction. Second enzyme, primase. Primase is an enzyme that synthesizes the primer. Okay, primer. As I said, primer is a RNA sequence, right? It is a RNA complementary to DNA. Okay, so this primase can also be said as RNA polymerase. RNA polymerase. Okay, synthesis of primer. Function is synthesis of primer. Then the third important enzyme is the helicase. Also said as DNA helicase. Okay, helicase function is unwinding. Okay, unwinding of DNA. As I said, firstly, DNA is bound to each other, right? Then it causes a bulging like thing. Okay, bubble. The bonds are broken. So this takes place because of helicase, as well as this thing, formation of replication fork. This replication fork. This thing. The bonds are broken by the helicase. Helicase will move from, see, we can imagine this is a ring-like structure. It moves from one strand, right, in one direction, and the bonds are broken. Okay, and the what we can say, replication fork is formed. The helicase function is unwinding of DNA, right, or we can say DNA melting. Then the next enzyme is the single stranded binding protein. Single strand binding protein. Okay, this protein is as a stabilizing protein. Okay, stabilizing function it has. It stabilizes this bond, stabilizes the strands which are broken by helicase. Right, suppose this is a protein, it will get attached to all the strands here. If one gets attached, they call other strands, other proteins to come in contact here, stabilizing the single strand so that it does not bind to each other, right? And again, double stranded is not formed. So to remain in the single strand state, binding protein gets bind to single strand and prevents rebinding of the unwind strands, right? Then there is another enzyme known as topoisomerase. Topoisomerase. Topoisomerase, see, topoisomerase means what? DNA structure is having its own topology, or we can say strains on it, right? Suppose as the replication fork proceeds, thus DNA strand gets supercoiled. Okay, here the terms come, supercoiling. So supercoiling means DNA structure gets strain on it. Okay, so to prevent this means to continue the DNA replication without getting strain on it. Okay, to keep the DNA structure in relaxed state, or we can say relaxed conformation, right? Relaxed structure. Topoisomerases are recruited there to prevent this supercoiling. Okay, means tension on the DNA structure is prevented by the topoisomerases. Again, in topoisomerases, there are types. RNase H. RNase H. See, RNase H is required for the primer removal. Okay, RNase H. See, primers are RNA. RNA nucleotides are there. So those RNA should be removed from the DNA strand because we are using RNA primers for free 3' OH group only. Okay, and thus it is not important for the, as such, it should not be remained in the DNA structure. It should be removed out. So to remove this primers, RNase H work. Ligase, DNA ligase, or you can say ligase. Ligase just joins the DNA strands to each other. Okay, if there is a nick, means if there is a break, slight break in the DNA strand. Suppose I draw here a structure like this. You can see here a small gap here. This gap will be filled by ligase. Ligase means joining. Okay, joining the two strands of DNA is brought about by DNA ligase. Last enzyme is nuclease. Nuclease. The nuclease are of two types, endonucleases and exonucleases. Okay, endonucleases and exonucleases. Here comes a term known as proofreading. Proofreading is a term. So what is proofreading? What happens during DNA replication? Some incorrect nucleotides are added. Okay, incorrect nucleotides are added which are not complementary to the template. Okay, we can say like that nothing is perfect, right? There are some errors. So those incorrect nucleotides are removed by this enzymes. Nuclease are the enzymes which degrade nucleic acid, okay, or remove the nucleic acids, right? We will discuss this more detail in the next slide. See, exonuclease. Exonuclease remove nucleic acids from the outside. Okay, means what? See, we know this is a strand and this is a synthesizing strand. Okay, 3 prime to 5 prime and 5 prime to 3 prime. Okay, so this strand will grow like this. Now here, here there is correct nucleotide. Now I will change the color for nucleotide here. So this is an incorrect nucleotide. Incorrect nucleotide. So this incorrect nucleotide has to be removed. So this is a 3 prime end. So here comes the work of 3 prime exonuclease. Okay, 3 prime exonuclease. So we can see these are types 3 prime to 5 prime exonuclease and 5 prime to 3 prime exonuclease. Endonuclease means it will go at the specific site within the segment and remove the nucleotide. Okay. Endo means it will go inside directly. Exo means it will cut by outside only. This all enzymes are important. So these are the general names actually. In eukaryotes, or we can say in prokaryotes and eukaryotes, the names of these polymerases or the enzymes are different, right? We can say these are the common names, but the specific names are different in species to species, or we can say organisms to organisms. So that we will discuss from the next videos. Okay, specifically talking, we will first discuss in detail about DNA polymerases because these are the very important enzymes for the application. So next video will be on the DNA polymerases types and their functions. Rest enzymes I have been covered in this video. The basic structure of each of the enzyme, the working that we'll discuss as we'll go to the process. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever the video is out. Also, follow us on all our social media pages and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.